Hi everyone. I'm going to be talking about error-prone PCR, what we've previously learned, and some of the strengths and weaknesses associated with it compared to other methods of library design. So error-prone PCR is a method for random mutagenesis. Template DNA from the protein of interest undergoes PCR with Taq polymerase, which provides a base error rate. To increase this rate, magnesium and manganese ions can be incorporated to stabilize nonspecific pairing, along with 8-oxo-DGTP and DPTP. As well, additional cycles can be run to increase the number of errors. By altering these parameters, we can effectively tune the error rate for our application. Due to the nature of this technique, a large quantity of mutants can be created with relatively broad diversity. As well, due to the presence of the template DNA in our mixture, we can ensure that our wild type is also included in the library. But why do we care about error-prone PCR? Why can't we just use computational techniques, rational design, or degenerate codon libraries like NNK? Well, often we lack structural information, either about the protein or its target. This makes computational design either difficult or impossible. The lack of information could also make NNK libraries impractical due to the large number of mutants generated per residue. Error-prone PCR creates a middle ground that samples a diverse region of sequence space while conserving most of the protein and limiting the library size. Error-prone PCR is often advised either when using a wild-type protein with relatively little structural knowledge or for a protein that has had difficult evolving by other methods. Error-prone PCR provides a shotgun approach that can access previously unsampled areas of sequence space in wild-type proteins or escape libraries trapped in a local maximum of binding affinity, as shown to the right. Effectively, it can jump from dot to dot to provide more favorable starting location for screening. Also, higher mutation rates promote larger increases in activity by finding rare mutations not previously incorporated. As well, error-prone PCR acts away from the binding site and can identify framework mutations that may provide stability. Finally, error-prone PCR is a quick and relatively inexpensive technique compared to primer or computational methods. While error-prone PCR can produce excellent results, it is not indicated for all uses. While it can provide large leaps in activity, the majority of mutants produced by this method are non-active, with a fraction of functional mutants decreasing exponentially with increasing error rate. Error-prone PCR also provides diminishing returns with libraries approaching the global binding maximum, where its broad diversity has a detrimental effect. An additional concern is that error-prone PCR does not include the control of other methods. Property changes, such as promiscuity, excess charge, or altered secondary or tertiary structure can accidentally be introduced into the protein. As a parting note, I'd like to address the misnomer of random mutagenesis. Often, error-prone PCR is not truly random. For one, polymerases favor specific nucleotide substitutions, and depending upon the location of the mutation in the codon, a change in genotype may not transfer to a change in phenotype. Complicating this further, multiple substitutions in a single codon are increasingly unlikely, making the number of mutations that cause a residue change even smaller. When a mutation does result in a residue change, the change is inherently biased by the distribution of codons, as shown on the right. This resulted in certain amino acids being underrepresented in libraries generated by this method. Well, I think I'm about time. Thanks for listening. I hope this was helpful, and I'll see you all in class.